Yeah, welcome to video series to understand all about metabolism of different life things. Now this topic is important because many questions are asked from this metabolism. The entire lipoproteins metabolism can be asked as a long essay. Apart from that, individual metabolism of chylomicron, VLDL, HDL, LDL can be asked either as a 5-marker question or a 3-marker question, and apolipoproteins and their function can be asked as 5 or 3-marker question. And from patient point of view, this in this Frederickson's hyper and hypolipoproteinemia is of clinical significance. So this is the most common question asked: enumerate lipoproteins and list their functions. It can be asked as a 3-marker or 5-marker question. So these are the important lipoproteins you need to remember: chylomicrons and apolipoprotein A1, B48, C2 and E are present on the chylomicrons, which facilitate transport of exogenous triglycerides. Very low density lipoprotein has got ApoB100, C2 and E and is involved in transportation of endogenous triglycerides. Intermediate density lipoprotein has got apolipoprotein B100 and E on its surface and is involved in transportation of cholesterol from liver to peripheral tissue. Low density lipoprotein has got ApoB100 on its surface and is involved in transportation of cholesterol from liver to peripheral tissue. So the last one is high density lipoprotein. This has got apolipoprotein A1, C2E on its surface and is involved in transportation of cholesterol from peripheral tissues back to liver. So this is also known as liver transport. Now this is the overview of the entire lipoprotein metabolism. Let's start from this one. Which one? Chylomicron. Now these chylomicrons are synthesized in the intestinal epithelial cells, and this is known as nascent chylomicron. So whatever the dietary triglycerides we consume, this is known as exogenous triglycerides. They are packed into chylomicrons. Along with this, the free cholesterol which is present in the diet is esterified with the help of esterase enzyme to form cholesterol esters, and along with this the phospholipids all are packed into chylomicrons, and this has got ApoB48 on its surface. Now this nascent chylomicron is initially released into lymphatics because the size of the chylomicrons is very large, which will not fit into the size of the capillaries. So it is initially released into lymphatics, and through subclavian vein it enters into general circulation. Now so it gets now the HDL2 transfers apolipoprotein C2 and E to nascent chylomicron, so that it gets converted to mature chylomicron. Now this mature chylomicron, this C2 will activate the lipoprotein lipase that is present in the endothelial cells of the blood vessels, and this will hydrolyze the exogenous triglyceride carried by chylomicron to fatty acid and glycerol. Glycerol is taken up by liver, and fatty acids are taken up by the skeletal muscles, wherein it gets oxidized and provides the energy. And this mature chylomicron, after losing the triglycerides, get converted to chylomicron remnant with the presence of cholesterol esters in its core, and it has now got only ApoE. Now with the help of these ApoE receptors, now these ApoE receptors are present on the surface of the hepatocytes. So this chylomicron remnant is taken to hepatocyte, wherein the cholesterol ester is released. This is acted upon by hepatic lipase, and the free cholesterol is generated in the hepatocytes. So this is about the metabolism of chylomicron. Now let's understand the metabolism of VLDL, IDL and LDL. Now, inside the hepatocytes, there occurs denosynthesis of fatty acids from acetyl-CoA. Now, these fatty acids are esterified with glycerol to form triglycerides. Similarly, denosynthesis of cholesterol occurs inside the hepatocytes. Now, this is again esterified with fatty acids from cholesterol esters. These two are packed into very low density lipoprotein, and this has got apolipoprotein B100s on its surface. Now, this is known as nascent VLDL, which is released into circulation. Now this nascent VLDL receives apolipoprotein C2 and E with the help of cholesterol esterase transport protein, and gets converted to mature VLDL, which carries endogenous triglycerides and cholesterol esters, and now it has got ApoB100, E and C2 on its surface. Now this apolipoprotein C2 activates lipoprotein lipase, which is present in the endothelial cells of the blood vessels, and this will hydrolyze the endogenous triglycerides to fatty acids and glycerol. So the glycerol goes back to liver, and fatty acids are taken by the skeletal muscles, wherein they get oxidized and provide energy to the muscle contraction. Now, so after losing the triglycerides, now this VLDL gets converted to intermediate density lipoprotein, which has now got only cholesterol esters in its core, and it has got ApoE and B100 on its surface. Okay. Now there are with the with ApoE, so it can be taken up by the hepatocytes. But majority of the IDL get converted to LDL. So wherein it loses its after it loses its ApoE, so it gets converted to low density lipoprotein, which has got only now ApoB100 on its surface. Now this LDL delivers the cholesterol esters to all the peripheral tissue. So in the peripheral tissue, so what happens, esterase enzyme hydrolyzes cholesterol esters to free cholesterol. Now this cholesterol is utilized for so many purposes inside the peripheral cell depending on its specificity. So the cholesterol become component of the biomembrane. In skin, cholesterol acts as a precursor of vitamin D. It acts as a precursor of estrogen in ovary, progesterone during gestation, and it forms testosterone in testis, and it forms glucocorticoids and mineralocorticoids in adrenal gland. So whatever the excess cholesterol that is present in the peripheral tissue is esterified with the help of monounsaturated fatty acid with the help of the enzyme acyl cholesterol acyl transferase, and forms cholesterol ester, and this gets stored in the peripheral tissue. So this is about the metabolism of VLDL, IDL and LDL. Now let's understand about the metabolism of HDL. Now HDL is synthesized in intestine also, and some amount of HDL is also synthesized in hepatocytes. Okay, so this nascent HDL has got mainly phospholipid, and ApoC2 and E are the apolipoproteins that are present on the surface of the nascent HDL inside the enterocytes. Now they are released into the general circulation, so they go to peripheral tissue and collects the cholesterol. Okay, so after collecting the cholesterol, A1 and A2 apolipoprotein get attached to this HDL, and this is known as HDL3 fraction or subfraction. Okay, now this cholesterol that is present in HDL3 is esterified with the help of polyunsaturated fatty acid with the help of the enzyme lecithin cholesterol acyl transferase, and this enzyme is activated by ApoA1 present on the surface of subfraction HDL3. So once the cholesterol undergoes esterification to form cholesterol esters, HDL3 gets converted to HDL2. Now this has got ApoC2 E and ApoA2 on its surface. This apolipoprotein C2 and E are transferred from HDL2 to nascent VLDL and nascent chylomicron to make them mature, and some amount of cholesterol esters are transported from the HDL2 to mature chylomicron and mature VLDL with the help of cholesterol ester transport protein. So the remaining part of this HDL with contains cholesterol esters in its core gets transported to liver. So now with the help of this scavenging receptor 1, which are present on the surface of the hepatocytes, this cholesterol ester from HDL2 is released inside the hepatocytes. This is acted upon by hepatic lipase, which will release the cholesterol, and this cholesterol gets converted to bile acid. So this was about the overview of the entire lipoprotein metabolism. So this can be asked as apart from that, each metabolism of this individual lipoprotein can be asked as a 5 marker. Now just to have a quick summary, the exogenous triglycerides or the dietary triglycerides are packed into chylomicrons, and they are the nascent chylomicrons which has got ApoB48 and A. They are released through lymphatics into general circulation. Now with the help of transfer of ApoC2 from HDL, it gets converted to mature chylomicron, and the C2 activates lipoprotein lipase, which will hydrolyze the triglycerides. So the fatty acids are taken up by the skeletal muscle, where it undergoes beta oxidation to provide energy for the muscle contraction, and so excess of fatty acid goes to adipose tissue, where it combines with glycerol, forms triglyceride and gets stored. So this glycerol goes back to liver, and this glycerol again can be used for esterification. They combine with fatty acid, can form TG, or it can be through dihydroxyestrogen phosphate, it will enter into glycolytic pathway. So this mature chylomicron, after losing its TG, gets converted to chylomicron remnant, and with the help of apolipoprotein E, it is taken up by the hepatocytes, and the cholesterol esters are released. Cholesterol esters are acted upon by hepatic lipase enzyme, releasing the cholesterol. So when only chylomicron metabolism is asked, you can write this. So a quick summary of metabolism of VLDL, IDL and HDL. Now the acetyl-CoA is the starting material for denosynthesis of fatty acids and denosynthesis of cholesterol. Now these fatty acids combine with glycerol to form endogenous triglycerides and cholesterol esterified fatty acids to form cholesterol esters. They are packed into very low density lipoproteins with ApoB100 and E on its surface. Now this becomes a nascent VLDL. Now this receives apolipoprotein C2 and E from HDL2, so it gets converted to mature VLDL. Now this mature VLDL, the C2 that is present on the surface of the mature VLDL will activate the lipoprotein lipase that is present in the endothelial cells of the blood vessel. So this will hydrolyze the endogenous triglycerides to fatty acid and glycerol. So as you all now know, fatty acids are taken up by the skeletal muscle where it undergoes beta oxidation to provide energy, and glycerol is taken up by the hepatocytes. Okay. Now this VLDL gets converted to intermediate density lipoproteins with ApoB100 and C on its surface. Okay. So now this can be taken up by the E receptor that are present on the hepatocytes. So once the remaining part gets converted to low density lipoproteins, which has got only cholesterol esters with ApoB100 on its surface. Okay. So once it loses its triglyceride, endogenous triglyceride content, so this VLDL gets converted to VLDL remnant, and through apolipoprotein E, it goes and binds to the E receptors on the surface of the hepatocytes, releasing the cholesterol esters, which is acted upon by hepatic lipase, releasing the free cholesterol. So this is about metabolism of VLDL, IDL and LDL. Okay. So coming to metabolism of LDL. So that the LDL that is formed from VLDL and IDL, so gets transfers the cholesterol to all the peripheral tissues. Okay. Now this through ApoB100 receptors which are present on the peripheral tissues. So this releases the cholesterol esters into the peripheral tissues. So this cholesterol esters is acted upon by esterase enzyme, releasing the free cholesterol. Now this free cholesterol becomes component of biomembrane. It acts as a precursor of vitamin D in skin. It acts as a precursor of steroid hormone, especially estrogen in ovary, testosterone in testes, glucocorticoids and mineralocorticoids in adrenal gland, and it also provides insulation to all the nerves of the nervous system. So excess cholesterol is esterified with monounsaturated fatty acid with the help of enzyme acyl cholesterol acyl transferase to form cholesterol ester, and this gets stored in all the peripheral tissue. Further excess of cholesterol is taken up by the macrophages, and these macrophages get converted to foam cells, and foam cells form the atheromatous plaque. So when the plaque expands, it will obliterate the lumen of the blood vessel, resulting in complete obstruction of the blood vessel, and it can result in infarction of any organ. Okay. So it can involve the major medium sized blood vessels or the large sized blood vessels, and when the blood vessels of the cardiac tissue are involved, it results in myocardial infarction, and when blood vessels of the brain are involved, it can result in cerebral infarction and can manifest as stroke, and when the vessels of the lower limbs are involved, it can result in peripheral arterial disease. Okay. This is about the significance of low density lipoprotein. Okay. So the question can be asked as describe of LDL. Coming to summarization of HDL metabolism, and the question asked is describe metabolism of HDL. Straightforward question, 5 marker. Okay. Now the HDL is the nascent HDL can be synthesized both in the hepatocytes and also in the intestinal epithelial cell, which is discoid in shape and has got apo C2 and E on its surface. Okay. So now this nascent HDL goes to peripheral tissue where it gets collects the cholesterol from the peripheral tissue, and once it collects the cholesterol, apo A1 and A2 gets attached onto the surface of the HDL, and it becomes subfraction HDL subfraction 3. Okay. Now this cholesterol is esterified to cholesterol ester with the help of, and the esterification detoxification is done by polyunsaturated fatty acid, and the enzyme is lecithin cholesterol acyl transferase, and this is activated by ApoA1 that is present on the surface of the HDL3. So once the cholesterol esters are formed, HDL3 gets converted to HDL2. Now this HDL2 normally transfers ApoC2 C2 to nascent VLDL and nascent chylomicron to become mature VLDL and mature chylomicron. Okay, and it also transfers the cholesterol esters to nascent VLDL and nascent chylomicron, so that the cholesterol esters are carried by VLDL and chylomicron also. Okay, now this HDL2 is taken up by the hepatocyte with the help of the receptors, SRB1 receptors, and it releases the cholesterol esters inside the hepatocytes. This is acted upon by hepatic lipase enzyme, releasing the free cholesterol, and this cholesterol is utilized for the formation of. Yeah, coming to hyperlipoproteinemias. Now, type 1 is characterized by deficiency of lipoprotein lipase, which is involved in hydrolysis of endogenous and exogenous triglycerides carried by VLDL and chylomicron respectively. So, this results in accumulation of mainly triglycerides. Type 2, we have got A and B. Type 2A is characterized by defective ApoB100 receptor. So, what happens is there occurs accumulation of LDL because LDL cannot cannot deliver the cholesterol to the peripheral tissue due to abnormal ApoB100 receptors. Now type 2 is characterized by excess of ApoB100 on the surface of LDL. So this also results in large amount of LDL in the blood circulation. So type 2A is ApoB100 receptor abnormality or deficiency. 2B is excess of ApoB100. Okay. Now type 3 is characterized by abnormal ApoE receptors. Now this through E receptors, all the lipoproteins are taken up by the hepatic tissues, so release their cholesterol. So this can result in accumulation of all this VLDL, chylomicron remnant, IDL, LDL. All this can be abnormally accumulated when there is deficiency of ApoE receptors, and this is known as type 3 hyperlipoproteinemia. Type 4 is characterized by increased production of VLDL inside the hepatocytes. Okay, so this is type 4, and we have one more variety, that is type 5. Type 5 always occurs secondary to some other disorders, mainly seen in metabolic syndrome, diabetes mellitus, characterized by abnormally high lipoprotein. Sorry, and type 5 is always secondary. Here also there occurs accumulation of VLDL and chylomicrons. Mainly the TGs, triglycerides are increased when lipid profile is done, and this usually occurs secondary to diabetes mellitus and ischemic heart disease. Okay, so these are the features. Coming to hypolipoproteinemias. So there are 4 in number. So familial hypo-beta lipoproteinemia. So this is characterized by decreased low density lipoprotein and decreased ApoB100. So this is actually good to our body because when low density lipoprotein is decreased, so this will act as some sort of protection to coronary artery disease. Then high A beta lipoproteinemia. So the B48, B100 both are decreased, and so there is decreased VLDL and LDL. So the total cholesterol also decreased, the triglycerides also decrease, and this usually is seen among malabsorption. Okay, so malabsorption is a condition characterized where there is defective absorption of this lipids and other biomolecules from the small intestine into general circulation. Okay, so hypo alpha lipoproteinemia. So this is characterized by decreased HDL and decreased ApoA1. The total cholesterol remains normal, triglycerides remain normal. As you all know, HDL is involved in reverse cholesterol transport. So if the HDL concentration decreases, so what happens? The cholesterol is not transported from the peripheral tissues back to liver for excretion. So this increases the risk of that particular individual to coronary artery disease. Similarly, familial alpha lipoprotein deficiency, it is also characterized by decreased HDL and A1. So total cholesterol remains normal, triglycerides remain normal, and this familial alpha lipoprotein deficiency has been observed in an island in Northwest Africa known as Tangiers Island. So this disease is named after that particular island where maximum individuals with familial alpha lipoprotein lipoprotein deficiency were encountered. So it's also known as Tangiers disease. So this predisposes all the young children to the development of coronary artery disease. The reason being the same, as HDL is involved in reverse cholesterol transport, because there is decreased formation of HDL in these individuals. So there occurs the cholesterol is not transported from the peripheral tissue and also from the blood vessels back to liver for excretion. It gets accumulated, predispose the patient to the development of coronary artery disease. So the other characteristic feature in Tangiers disease is the biochemical basis remains same. There is failure in transportation of cholesterol from peripheral tissues. It's not called peripheral tissues to HDL. So results in cholesterol accumulation, and the cholesterol can accumulate in tonsils, and these tonsils appear as large yellow tonsils. It can abnormally accumulate in the nerves, resulting in recurrent neuropathies, and predispose individuals to early onset of atherosclerosis and early myocardium. So that was all about the metabolism of lipoproteins. Coming to the segment part, which lipoprotein transports dietary triglycerides from intestine? Simple, chylomicrons, which are formed in the intestinal mucosa. They carry exogenous okay. Major apolipoprotein of chylomicron is B40. Okay, so lipoprotein lipase is activated by C2. Okay, now this C2 is donated to VLDL and chylomicron by HDL2. That you should. So lipoprotein lipase is present on endothelium of the capillaries, which will hydrolyze endogenous and exogenous triglycerides to fatty acids. Endogenous triglycerides are transported by VLDL. Major apolipoprotein of VLDL is intermediate compound formed from VLDL is IDL, intermediate density protein. Which lipoprotein delivers cholesterol to tissues? It is low lipoprotein. LDL receptor recognizes ApoB100, okay, which are present on all the peripheral tissues. Reverse cholesterol transport is mediated by HDL. Major apolipoprotein of HDL is ApoB. Okay, LCAT, lecithin cholesterol acyl transferase, is activated by ApoA1, which will esterify the cholesterol which is present in which is carried by HDL3 from peripheral tissues, and HDL3 after esterification of this cholesterol gets converted to HDL2, more dense lipoprotein. Okay. Function of LCAT is esterification of cholesterol. Which apolipoprotein helps hepatic uptake of remnants? It is E. Okay, so this will help uptake of VLDL remnant, chylomicron remnant, this IDL, LDL, everything is taken up inside the hepatocytes, where the cholesterol is delivered for excretion with the help of E. Now chylomicron remnants are taken up by deficiency of lipoprotein lipase leads to hypertriglyceridemia, because this is involved in breaking down of both endogenous and exogenous triglycerides. So increased triglycerides occurs when there occurs deficiency of lipoprotein lipase, which is Fredrickson's type 1 lipoproteinemia. Tangiers disease is due to deficiency of which enzyme hydrolyzes triglycerides in chylomicron? It is lipoprotein lipase. HDL transfers cholesterol to liver via SRB1 receptors. It is not E. E is for VLDL and chylomicron remnant, and also for IDL. For HDL, it is SRB1 receptor. You need to. Fredrickson's type 1 hyperlipoproteinemia is due to deficiency of lipoprotein lipase deficiency. Most atherogenic lipoprotein, as you know, is low lipoprotein. So that was all about metabolism of lipoprotein. Check out your MCQ score. If the score is more than 50% good, highly appreciable. Less, watch the rewatch the video and redo all the MCQs. So thanks for watching. So if you like the video, please do like, share and subscribe. So the next video will be on prostaglandins. Thank you. Happy learning.